Imagine you have just installed this massive state-of-the-art security system in your house.
Right. Totally impenetrable.
Exactly. You've got reinforced doors, unbreakable glass, and, motion sensors on literally every window. You feel completely safe.
As you should.
But what if a burglar could somehow send a remote, invisible signal to your house weeks before they ever even set foot on your property?
That is deeply terrifying.
Right. A signal that silently instructs your own security system to, slowly unlock the doors, disable the alarms, and just prop open the windows.
So by the time they actually show up, there's no breaking and entering.
None at all. They just stroll right through the front door.
It's a profoundly unsettling concept, the idea that your own defenses are actively preparing the way for an invasion.
Yeah, you're completely compromised from the inside out, and not a single alarm bell rings until the damage is already done.
Which is usually too late.
And as wild as that science fiction scenario sounds, we are going to explore how that exact process is happening right now at a microscopic level inside the human body.
It really is happening.
Welcome to another deep dive. Today, we are unpacking a really groundbreaking 2026 study from the journal Immunity.
A fantastic paper.
It really is. Our mission today is to trace an unbroken, almost diabolical molecular chain reaction. We are looking at triple-negative breast cancer, which is frequently referred to as TNBC.
Right, TNBC.
This research reveals how a tumor in the breast literally hacks the blood vessels in the lungs to roll out a biological red carpet, ensuring the cancer can spread.
It's wild.
But we are also going to look at the counterattack. We will explore how a very famous class of metabolic drugs, ones you usually hear about for, diabetes and weight loss, might be the ultimate key to changing the locks and keeping those doors shut tight.
The clinical stakes for this specific type of cancer, TNBC, really cannot be overstated.
Yeah, it's incredibly aggressive, right?
Notoriously aggressive. It lacks the 3 common receptors that we typically target with conventional hormone therapies.
So the usual treatments they don't work as well.
Exactly, making it incredibly difficult to treat. But what makes TNBC so uniquely deadly is its high propensity to metastasize or, spread, specifically to the lungs.
Okay, let's unpack this, because for decades, the prevailing thought was that cancer cells just sort of break off, right?
Right, they float aimlessly through the bloodstream, happen to land in the lung, and just try to survive.
But this paper shatters that assumption.
Completely shatters it. Long before the cancer cells even leave the breast, the primary tumor is chemically communicating with the lung.
It's sending that remote hacker signal.
Yes. It is preparing what oncologists call a pre-metastatic niche.
A PMN.
Right. It is actively fertilizing the soil miles away from the source.
Okay, so let's zoom way in on the mechanics of that, because how a tumor in the breast alters the soil of the lungs is just a fascinating mystery.
It really is.
To understand it, we have to look at the blood vessels inside the lungs. These tiny capillaries are lined with these specialized structures called endothelial cells.
If we stick with your house analogy, think of endothelial cells as the literal bricks making up the exterior walls of the blood vessel.
Okay, the cellular bricks.
Right. In a healthy lung, these cellular bricks form a remarkably tight, leak-proof barrier. They allow oxygen to pass through into your bloodstream, but they ensure that the cells and fluids in the blood stay contained.
Keeping the bad stuff out.
Exactly. During the progression of TNBC, however, these endothelial bricks receive that remote signal we mentioned from the primary breast tumor.
The hacking code.
Yes. This remote signal forces the lung's endothelial cells to dramatically overproduce a specific enzyme called FASN.
Okay, FASN. What does that stand for?
That stands for fatty acid synthase.
So FASN is essentially a molecular factory.
A literal fat factory.
When the cancer forces this enzyme into overdrive, these lung endothelial cells undergo a massive shift. They just start mass-producing fat.
And specifically, they use a biological process called de novo lipid synthesis.
De novo meaning from scratch, right?
Exactly. They are building fat molecules from scratch to pump out a very specific saturated long-chain fatty acid called palmitic acid.
And this behavior is, highly abnormal, isn't it?
Extremely abnormal. Most adult human cells do not build fat from scratch. They simply pull the circulating fats they need from our diet out of the bloodstream.
But in this scenario, the lungs' blood vessels are transformed into unauthorized palmitic acid factories.
Right, secreting massive amounts of this specific fat directly into the surrounding lung tissue.
The evidence supporting this in the source material is just wild to read.
The mouse models they used were incredible.
Yeah, they mapped this out using specialized mouse models. specifically the 4T1 and AT3 tumor models.
And what they found was striking.
They observed that triglycerides and free fatty acids selectively accumulated in the lungs of these mice.
Only the lungs.
Right. The fat didn't build up in the brain. It didn't build up in the liver. It was exclusively targeting the lungs.
And to push the hypothesis even further, they ran dietary experiments.
Oh, this part is crazy.
They fed some mice a high-fat diet and others a high palmitic acid diet.
Literally food enriched with the exact fat the FASN factory produces.
Exactly. And the results were grim. The mice on the high palmitic acid diets experienced a massive explosion in lung metastasis.
And a severe drop in overall survival.
It was a very clear correlation.
But wait, let me push back on this for a second because I want to make sure the logic tracks for you listening.
Sure, go ahead.
Palmitic acid is just a common saturated fat. It's in palm oil, it's in red meat, it's in dairy. We eat it, like, all the time.
We do.
How does a simple dietary fat cause cancer to spread so aggressively? Is it just acting as a supercharged food source?
Like feeding a fire?
Yeah, like, are we just feeding the cancer cells so they grow faster when they arrive?
That is the most intuitive assumption, and, well, it is partially true.
Okay.
The interstitial fluid in the lung, which is the liquid space between the cells, is actually very deficient in glucose.
And cancer cells typically love sugar, right?
Exactly. But when they arrive in the lung, they are forced to switch their metabolism. They have to burn fat for energy instead.
Oh, wow. So the palmitic acid does act as a crucial fuel source.
It does. But if we connect this to the deeper mechanism the paper outlines, its role as a fuel is actually secondary.
Wait, really? Secondary to what?
Its much more sinister role is acting as a dangerous signaling molecule.
The text message.
Yes. It is essentially a corrupted piece of code, a hacked text message sent directly to your immune system, giving it terrible, destructive instructions.
Let's crystallize this first phase before we move on to the immune system, because the mechanism is dense, but it's super critical.
A quick recap is a great idea.
Let's do a mini recap. Bullet 1. First, the primary breast cancer sends a signal that forces the blood vessels in the lungs to overproduce the FASN enzyme.
Right.
Bullet 2. That FASN enzyme acts as a localized factory, building palmitic acid from scratch. And bullet 3. That palmitic acid builds up in the lung tissue, creating a highly fertile, fat-rich, pre-metastatic niche for the cancer to thrive in.
You nailed it.
Which brings up a really fascinating question for you listening to ponder. Think about your own daily diet for a second.
It changes your perspective completely.
It really does. We often think of the fat we eat purely in terms of, calories or heart health. But how does knowing that specific dietary fats can act as literal molecular text messages, instructing your immune system to do things, change the way you view a high-fat meal?
It definitely forces a paradigm shift in how we understand nutrition. Food isn't just energy, it's information.
And in this case, bad information.
Very bad. And the information carried by palmitic acid is intercepted by the body's first responders, the neutrophils.
Ah, the guard dogs. This is where the story takes a dark turn.
A very dark turn.
Normally, neutrophils are the unquestioned heroes of your immune system. If you get a paper cut and bacteria enter the wound, neutrophils are the first ones to rush to the scene.
Right, they engulf the invaders and destroy them.
But the study shows that when these neutrophil guard dogs enter the lung and find themselves bathed in this palmitic acid, they undergo a bizarre transformation.
They essentially get brainwashed by the fat.
Brainwashed guard dogs. Unbelievable.
The mechanistic breakdown of how this brainwashing occurs is it's a dark marvel of biology.
How does it actually happen?
The palmitic acid physically binds to a specific receptor on the outer membrane of the neutrophil called TLR4.
TLR4, coal-like receptor 4.
Exactly. You can conceptualize TLR4 as a motion sensor on the outside of the cell.
Okay, emotion sensor.
Under normal circumstances, this sensor is tuned to detect the presence of bacterial infections.
But the fat tricks it.
Yes. Palmitic acid fits the lock just well enough to trick it. Once triggered by the fat, this TLR4 motion sensor activates an internal relay wire.
And what's the wire called?
It's a protein called MyD88.
MyD88. So we're tracing the electrical wiring of the cell here.
Right, step by step.
The palmitic acid trips the TLR4 motion sensor on the outside, which sends an alarm signal down the internal MyD88 wire.
And that wire leads straight to the command center.
Right, to the master dispatcher.
Exactly. The signal travels down the wire and phosphorylates or activates a protein complex known as NF-kappa B P65.
NF-kappa B.
You can think of NF-kappa B as the 4-star general of the cell.
The guy calling the shots.
Once it receives the signal, this dispatcher marches straight into the nucleus of the neutrophil, binds to the DNA, and screams, "Start producing weapons."
And the weapons these brainwashed neutrophils start churning out are devastating.
They really are.
They start spewing massive amounts of a specific protein called lipocalin-2, or LCN2 for short.
LCN2, remember that name.
And at the same time, they start forming these structures called nets, which stands for neutrophil extracellular traps.
And nets are literally what they sound like.
Yeah, the neutrophil extrudes its own DNA, laces it with highly toxic tissue-degrading enzymes, and throws it out like a sticky spider web.
It's a brilliant but destructive defense mechanism.
So our loyal guard dogs get corrupted by the palmitic acid, and they start throwing out sticky, toxic webs and flooding the area with this LCN2 protein.
Which is basically an acid.
But I have to ask, what is the LCN2 actually targeting? I mean, is it accidentally hurting the cancer cells?
What's fascinating here is that the LCN2 doesn't harm the cancer at all.
Not at all.
No, it is weaponized against the body's own infrastructure. Specifically, it targets the blood vessels.
The very walls we need to protect us.
Exactly. If we return to our analogy of the endothelial cells being the bricks of the blood vessel wall, we have to remember that bricks alone don't make a waterproof wall.
Right, they need mortar.
They need a mortar to hold them tightly together. In biology, that mortar is made of tight junctions.
Tight junction.
And the most vital protein making up those tight junctions is called ZO1.
Zona occludens 1. Oh, wow. So the LCN2 protein is essentially an acid that targets the ZO1 mortar.
That is exactly the mechanism. LCN2 degrades the ZO1 tight junction proteins.
So the mortar dissolves.
As the mortar dissolves, the endothelial bricks loosen and pull apart.
The wall collapses.
The structural integrity of the capillary fails. The blood vessels become hyperpermeable. They become leaky.
And that is the exact moment the burglar simply opens the door.
The door is wide open.
Circulating tumor cells, the cancer cells that are broken off from the main breast tumor and are desperately floating through the bloodstream looking for a home, suddenly find these microscopic cracks in the leaky blood vessels.
They undergo a process called extravasation.
Extravasation.
Yeah.
They slip out of the blood vessel through the degraded mortar and invade the rich, fatty lung tissue waiting on the other side.
And we must emphasize that this isn't just an isolated phenomenon observed in mice.
Right. The human data.
The researchers rigorously cross-referenced their findings with human clinical data. They utilized the Cancer Genome Atlas, which is a massive database of human cancer genetics.
What did they find?
They discovered that human patients with triple-negative breast cancer consistently exhibit highly elevated levels of this LCN2 protein.
The same exact protein.
Furthermore, when pathologists examine actual tissue samples from human breast cancer patients who suffered lung metastases, they observe a striking pattern.
The co-localization.
Yes, heavy co-localization of neutrophils. And that activated NF-kappa B dispatcher right there in the lung tissue.
It's all connected.
The exact mechanism we are tracing, the brainwashed neutrophils destroying the vessel walls, is mirrored flawlessly in human patients.
It's a heavy reality to sit with.
It is.
But there is a deep empowerment in finally understanding the exact mechanical blueprints of the enemy. Let's do another quick recap. Let's synthesize this second phase of the chain reaction so we can see the whole picture before we talk about how to stop it.
Great. Let's run through it.
Bullet 1. The palmitic acid binds to the TLR4 motion sensor on the outside of the neutrophil, which activates the NF-kappa B dispatcher inside the cell.
Right.
Bullet 2. The dispatcher orders the neutrophils to release that LCN2 protein in those sticky DNA webs.
Weapon.
Bullet 3. The LCN2 acts like an acid, dissolving the ZO1 mortar between the cells of your blood vessels, rendering them leaky.
And bullet 4.
Exactly. Bullet 4. The circulating cancer cells slip through those leaks and colonize the lung.
A perfect summary.
So I have another question to challenge your thinking as you listen to this. If our own immune system can be tricked into tearing down our biological walls, how do we logically intervene? Do we fight the cancer directly, or do we try to put our guard dogs back on a leash?
That was the exact dilemma the research team faced.
Which way do you go?
Their first instinct was to cut the problem off at the root. If the FASN enzyme factory is producing the palmitic acid, why not just shut down the factory?
Makes total sense.
They tested a drug called orlistat, which is a known FASN inhibitor.
And reading this study, here's where it gets really interesting, because orlistat actually worked.
It did work.
It successfully stopped the FASN factory, lowered the levels of palmitic acid in the lungs, and significantly reduced the lung metastasis. But there was a massive catch that made it clinically unviable.
The catch is collateral damage.
Collateral damage.
Orlistat is a systemic drug. It affects the whole body and is actually used clinically as an anti-obesity medication.
Oh, right.
When given to the mice, they experienced severe systemic weight loss.
Which is bad.
In the context of a fragile cancer patient, inducing severe weight loss, or cachexia, is incredibly dangerous. It weakens the patient too much to endure further treatments.
So they realized they couldn't use a systemic shotgun approach. They needed a molecular sniper rifle.
Precisely. And enter the GLP-1 receptor agonists.
Yes. If you pay attention to medical news at all, you have absolutely heard of these. Drugs like dulaglutide and liraglutide, famous globally right now for revolutionizing the treatment of type 2 diabetes and obesity.
They're everywhere in the news.
But on the surface, using a blockbuster diabetes drug to stop breast cancer from spreading sounds like a typo. Why would they try this?
It stems from a brilliant piece of biological mapping. The researchers analyzed the lung tissue and made a critical discovery.
What was it?
Those specific endothelial cells, the bricks of our lung blood vessels, naturally express high levels of GLP-1 receptors on their surface.
Oh, so they already have the locks.
They realized they had a direct, built-in doorway. They could use these GLP-1 drugs to target the lung's blood vessels directly, bypassing the rest of the body.
And the results were just stunning. When the GLP-1 drug binds to those receptors on the lung endothelial cells, it directly downregulates. It shuts off the FASN factory.
It cuts the power.
Let's trace how this reverses the entire chain, because this is the ultimate aha moment of the entire deep dive. Follow the dominoes falling backward here.
This is the best part.
Okay, because the drug shuts down FASN, no palmitic acid is produced.
Right.
Because there is no palmitic acid, the neutrophil motion sensors are never tripped, and the cells stay calm.
They stay good guard dogs.
Because the neutrophils are calm, no LCN2 acid is ever released.
The mortar is safe.
Because there is no LCN2, the Z01 tight junctions, the mortar stay perfectly strong.
And because the junctions are strong,
the blood vessel walls remain sealed tight. The circulating cancer cells are completely trapped in the bloodstream, unable to break into the lung, where they eventually die off.
It is an incredibly elegant blockade. Notice that you aren't actually attacking the tumor cells directly with this drug.
Right, you're not trying to kill the cancer with the diabetes drug.
No, you are simply refusing to let the tumor alter your soil. You are maintaining the integrity of your own walls.
I do want to play devil's advocate here, though, because, as you mentioned, GLP-1 drugs are literally famous right now for causing dramatic weight loss in humans.
They are.
If the previous drug, orlistat, stopped the cancer but caused terrible weight loss, how do we know the GLP-1 isn't doing the exact same thing? Like, are we sure we aren't just starving the cancer because the host body is shedding weight?
It is a vital question, and the researchers designed specific controls to isolate that exact variable.
Okay.
They proved that the anti-metastatic effect of the GLP-1 drug was absolutely independent of systemic weight loss.
Oh, wow. So it's not about the weight.
Not at all. The dosage and the specific molecular mechanism operated as a localized signaling block right on the blood vessels. But the benefits didn't stop at just sealing the walls.
There's more.
The GLP-1 therapy actually reshaped the entire immune landscape of the lung.
Yes. The paper details how it changed the actual types of immune cells hanging around the lung. When the palmitic acid was flowing freely, the environment was dominated by highly suppressive neutrophils.
The bad guys.
These corrupted cells express high levels of a protein called PD-L1. Now, PD-L1 is essentially a chemical off switch that puts the rest of the immune system to sleep,
allowing the cancer to hide in plain sight.
But once the GLP-1 drug shut down the palmitic acid factory, those PD-L1 suppressive cells dropped dramatically.
The chemical smokescreen cleared.
And in their place, the lung experienced a huge surge in CD8-positive T cells.
And CD8-positive T cells are the heavy hitters.
Yeah, if neutrophils are the guard dogs, the CD8-positive T cells are the elite assassins of the immune system. Their sole biological purpose is to hunt down and terminate abnormal or cancerous cells.
So the drug didn't just rebuild the wall, it actively brought the elite guards back to the perimeter.
The graded, hierarchical evidence they present to back this up is phenomenal.
The combination therapies.
Exactly. They didn't just test this GLP-1 drug in a vacuum to see if it worked on its own. They tested it the way a real oncologist would treat a patient.
Right, in combination therapies.
They combined dulaglutide with classic heavy-hitting chemotherapy drugs like gemcitabine and doxorubicin.
Standard of care drugs.
And they also tested it alongside modern immunotherapy, specifically anti-PD-1 antibodies.
And the results.
In every single model, Combining the GLP-1 drug with standard treatments drastically shrank the tumors, prevented the lung metastasis, and significantly prolonged the survival of the mice, compared to using chemo or immunotherapy alone.
Which is the exact benchmark we need for future clinical trials. Cancer is a notoriously adaptable disease.
It mutates around everything.
It mutates around single obstacles, exactly. Hitting it from multiple intersecting angles, using a metabolic drug to protect the blood vessels while simultaneously attacking the tumor cells with chemotherapy or unleashing the immune system with antibodies is the gold standard for creating durable, lasting treatments.
Let's do one final mini recap. Let's pull all of this together and crystallize this final therapeutic phase.
Sounds good.
Bullet 1. We learned that metabolic diabetes drugs, specifically GLP-1s, can bind directly to the blood vessels in the lungs, shutting down the FASN factory.
Cutting the power.
Bullet 2. By stopping the palmitic acid at the source, the destructive chain reaction is halted.
Right.
Bullet 3. The blood vessels stay sealed, and the immune system rebalances, bringing back those cancer-killing assassin T cells to finish the job.
Perfect.
So, a question for you to spark your curiosity. Can you think of other instances in medical history where a drug designed to fix one completely separate system, like a metabolic condition, turned out to be the biological silver bullet for a seemingly unrelated disease?
It really highlights how deeply interconnected the human body is.
Doesn't it? We like to put diseases in neat little boxes, but our biology just doesn't respect those boundaries.
The human body is a single, massively complex network. The metabolic system, the immune system, and the vascular system are not separate silos. They are engaged in a constant, lifelong chemical conversation.
So what does this all mean for the future of medicine? We've traced an incredible invisible journey today. We saw how a primary breast tumor acts as a remote hacker, sending signals to the lungs to produce the FASN enzyme.
We saw that FASN factory flood the lung tissue with palmitic acid.
We watched that fatty acid act as a corrupted piece of code, binding to the motion sensors of our own neutrophils, activating their internal dispatchers, and forcing them to spew out toxic webs and the LCN2 protein.
We saw that LCN2 melt the mortar holding our blood vessels together, allowing the cancer to slip inside.
But most importantly, we saw how we can use existing metabolic tools to act as molecular wire cutters, shutting down the factory and stopping the explosion before the fuse is even lit.
If there is one overarching philosophy to take away from this research, it is a fundamental paradigm shift in how we approach oncology.
A complete shift.
For decades, the war on cancer has been fought with a very localized search and destroy mentality aimed squarely at the tumor itself.
Burn it with radiation, poison it with chemotherapy, cut it out with surgery.
Exactly. But this deep dive provides overwhelming evidence that cancer is a disease of the entire ecosystem.
The soil matters.
A seed, no matter how aggressive, cannot grow if it lands on solid concrete. If we use metabolic interventions to deny the cancer its fertile soil, and if we protect the structural integrity of our blood vessels, the cancer simply cannot take root.
It is entirely about securing the perimeter, which brings us right back to our burglar at the beginning of the show. If the security system is uncompromised and the doors are locked, the burglar is just a guy standing on the porch with nowhere to go. But I want to leave you with one final provocative thought to mull over as you go about your day. We learned today that palmitic acid is the key signaling molecule that brainwashes the immune system to tear down our cellular walls in this specific cancer context.
Right.
We also know that modern Western diets are absolutely saturated with palmitic acid, very often derived from palm oil used in thousands of everyday processed foods.
It's in everything.
If high levels of this specific dietary fatty acid can act as a molecular instruction manual that literally tells your immune system to degrade your vascular integrity, what else is our daily food silently instructing our biology to do right now, at this very moment?
A very sobering thought.
Thank you so much for joining us on this dense, complex, and really fascinating journey through the molecular landscape of the human body. Stay curious, keep asking questions, and we will see you on the next deep dive.